get started today? We got 2 weeks left. 2 weeks from today, you'll be taking your final for this class. Holy cow. It's on Monday, 8 a.m. Next Monday is a Memorial Day holiday, so a couple days this week. We should wrap up our material this week and just have a review next week, so that'll be nice for you guys. What are these cells?
Sperm.
Sperm, good. How many chromosomes?
23.
23. All right, piece of cake. Organ. We'll get them. We got 2 weeks. Testes. Testes is the
Oh,
ovary.
it's the seminiferous vesicles.
Seminiferous tubules, interstitial cells, LH. Talked about that this morning. LH affects these to release testosterone. We left last week. I know we're bringing back, you guys have probably not been looking at this over the weekend, which is good. But this is the mother cell. We call that a spermatogonia. You guys remember that? 2n equals 46. You'll have to go through these because it's on the basilar wall out here. That's the spermatogonia, and as they develop through, we go type A, type B cells. And when we talk about primary spermatocytes, like these guys here, primary spermatocytes, that's 46. Then we get down to sperm or spermatid, and they have the tail, and then 23. So they go through that meiotic division as they're moving through. So a lot of questions can come off of this one. We'll get more comfortable with it as we move forward. Organ, uterine lining. All right, this is the endometrius of the uterus. We've got 3 phases, and that's what I'm talking about today. Is the hormones that affect proliferative, secretory, and then the menstrual phase of the endometrium of the uterus. And lastly, organ. Anybody know? This is the ovary, because this is the developing ovocyte surrounded by a zona pellucida, which I talked about today, corona radiata, which is the radiating crown, and then the antrum, which is fluid filled. This is all just about day 12 of the ovarian cycle, and it's about ready to be ovulated. So we'll go through all of these as we talk about the hormones today. Today we're going to just cover hormones. We don't really have labs left. We don't dissect an ovary or do anything like that. We have one model. We have a few slides that I just went through. So mainly lecture material. But as we're moving forward, I'm going to get through some genetics today as well. And so Wednesday, I'm hoping to finish genetics and introduce the endocrine worksheet to you, but then also have time in class to work on your genetics worksheet as a group or individually, because that's your extra credit. It's worth 5 points raw, so you have to do it. I can call it extra credit, but I want you all to do it and be successful. So we're going to have some time in class to work on that, and then we'll finish that up. It's due on review day, which I think we said was June 1. That's great if you weren't there. That'll be next Wednesday. All right, oogenesis. We talked about spermatogenesis, the making of sperm. Oogenesis is making oocytes or ovum. And oogonia are the diploid stem cell. That, if you don't remember that, diploid is 2n equals 46. You might put those numbers out there because it seems like a nice way for me to do a matching section where I have the number of chromosomes on one column and the different cell types on another column, and you have to tell me whether it's 23 or 46. So we start out with primary oocytes, and we start in the female. These are present at birth, and there's about 400,000 of them. It's crazy. They stay suspended until puberty at this primary oocyte stage. And at puberty, hormones start taking over. And that's no surprise to anybody in the class. One each cycle, if you will, is selected to go through meiosis 1, and we make a haploid secondary oocyte. And we get an odd cell. You guys remember when you have spermatogenesis, how many products, sperm, are formed from that? 4, all right, when you go through that development. But in the female, if you make 4 eggs, could you have 4 kids? Yeah, so we actually have these, what they call non-essential cells that are formed that are called polar bodies, and they're n equals 23. For years, polar bodies were never considered to be able to be fertilized because they didn't have enough cytoplasm. But now there are cases where we can have actually polar bodies being fertilized as well. And for many, many years, 100 years, polar bodies were just excess ways to remove our body of chromosome. Now they actually are finding they can be fertilized. So we'll talk about that as we go through. At this stage, we get a highly developed follicle called a Graphian or vesicular follicle. You can use either term, Graphian or vesicular follicle. And at this stage, the secondary oocyte is ovulating. This is day 14. We need to know the days of the cycle and what's going on in the days of the cycle. If it's penetrated by a sperm, it releases another polar body, and we complete meiosis too. You have all this in your notes, so I can kind of buzz through it. This is really important for your fun. The follicular phase, follicle cells develop. It's under the mainly under the control of estrogen levels. The zona pellucida is a glycoprotein around the oocyte as it develops. And then I showed you a picture this morning, and I told you about a radiating crown of cells called a corona radiata. Ovulatory phase. Ovulation occurs, secondary oocyte, that's N equals 23. Some women during ovulation experience a pain and can actually tell by physical pain in their abdomen that they're ovulating. And that's called, from German Mittel, Schmerz is pain, but middle pain, which is a German term for pain during ovulation. So some women can actually determine exactly when they're ovulating. And the ones that didn't, usually there's several that are starting to develop each cycle, and some of them don't make it to ovulation, and they have to be reabsorbed, and those are called atresia or atretic follicles, the ones that did not make it to ovulation. And then the luteal phase. Corpus luteum, double star this comes up on the test a lot. Corpus luteum, because it is making progesterone. That's the big one here. And it will turn into a corpus albicans. What's an albicans? Anybody know?
It's on the model.
It means white. It's on the model, that's right. And it's white. And that's degenerating tissue, scar tissue, if you will. Once there was a time in the medical community with autopsies determining the age of a female, they would actually dissect the ovaries and see how pitted and scarred it was. And that would give an indication of possible age based on the number of ovulations that had occurred. And it's all under hormonal control. Did you guys know that? All right, here's what you're looking at. This is the hardest part of the final exam. I try to make it as easy as possible, but this is a challenge. Because each month, we're using a month, 28 days, we go from this stage here all the way to here, all right? And so we are developing the oocyte. You'll notice the oocyte is staying about the same size. And we're going through this process, but the cells around it, which we just talked about in your notes, are called the follicle cells. So we've got an oocyte, and then we've got these granulosa or follicle cells that are surrounding them, and they're the ones that are actually growing. And we get out, so this is day 0 of the cycle, day 2, day 3, day 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and we're ready to ovulate, the secondary oocyte. And I told you how many chromosomes there? 23. Is there a specific hormone that is elevated that causes ovulation? Yeah, there is. It's called LH. All right, have you guys seen that yet? We're gonna get in the hormones next in your notes, and there's where it gets complicated, but I need to be able to ask you, at day 10, tell me about the follicle cells, and you'll say at day 10, the follicle cells are developing very quickly into this. Then, when ovulation occurs, we're left with this tissue here, which I just talked about as the corpus luteum, And this is releasing progesterone now. It's still important. This has been sent off. What picked it up? We did the anatomy last week. Remember the fingers? The fimbriae undulate, pick up the egg, and it's going to swing over here and get fertilized in the ampulla. Well, somebody needs to tell the uterus that the egg is on its way. And the progesterone coming from the corpus luteum signals the uterine lining to go into its secretory fluffy stage for implantation. If nothing happens, no fertilization occurs, it goes into scar tissue, and you're left with the corpus albicans, which is where we're at right now in the notes. You guys got all that? All right. And then we go back to day 0 again, and we start this again, perfectly 28 days. Not really. All right. Goes around like that, but you'll need to know and visualize that, because the next thing that we're going into in the notes is hormones, because hormones control all of these developmental phases. Yeah, Jason.
The lutein, what happens if fertilization does occur? What happens to it?
Fantastic question. That's why it comes up on the test a lot. Corpus luteum will maintain itself and release progesterone out to the uterus unless fertilization does not take place, and then it becomes the corpus albicans. If fertilization does take place and implantation occurs, this stays intact for another 3 to 4 months to maintain the uterine lining so that it doesn't slough off, and we can start to develop the embryo in the uterus. And then the placenta takes over the progesterone release function, and this will eventually degenerate anyway. But it does stay and maintain. That's usually a test question, Jason, so you guys might want to note that, is what happens to the corpus luteum if pregnancy occurs and it is maintained for several months. The hormonal cycle is complicated. It's similar to spermatogenesis. We start out here GnRH, and we release FSH and LH, and you'll notice FSH is on the top here, not LH, because we're not sure of LH's function at this point. But FSH is follicle stimulating hormone. What do you think it stimulates? Follicles, and these are follicle cells. And you'll notice as FSH is being released that the follicle cells are growing. And this is day 0, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way down to 14. FSH and LH. And then slightly elevated estrogen levels inhibits all of this. Then we get a lot of LH and FSH is being released. And this comes down later on. And this is LH now. LH causes ovulation, if you didn't know that. Is this on the endocrine worksheet? It sure is, all right. And LH in the female system does 3 important things. The major one that it does is that it causes ovulation. LH surging here to here, and then we have estrogen, progesterone, and inhibin that pause this process for about 14 more days to make sure and see if we have fertilization and implantation occurring. This is the feedback loop. You have to know the feedback loop of female hormones in order to understand this exam. And this is in your lab manual. It actually puts everything together, but you can see that we've got, these are over a day's period of time. There's our 4 major hormones for the female: LH, FSH, estrogens, and progesterone. And this is a day chart. 0 down here, 28 down here. You'll notice that progesterone is irrelevant until about day 14. That's because the corpus luteum is making progesterone and it is not present. Yeah, but estrogen is early, day 0 to 14, estrogen is very important. LH, FSH surge, LH causes ovulation, then they drop off, and then progesterone takes over to maintain the uterine lining for fertilization. Here's the big picture, and this comes out of your lab manual as well, it's also in your textbook. But we're talking about days over here. What's going on in the uterus? You'll notice it says uterine cycle and ovarian cycle. Oh, there's two? Yep, 2 cycles. Ovarian, you'll see what's going on here. Corpus luteum and degenerating. You'll see what's going on in the uterus. You'll see the phases of what's going on. So if I ask you, day 10, and I do this on the exam, day 10, what is the phase of the uterine lining? You'll go, well, it's proliferative. It's getting ready for implantation. What's the stage of the ovary? Well, you go up here, it's in the vesicular follicle stage. What's the level of progesterone at day 10? Can you put it all together? We'll go back to that. Day 10, right here. Level of progesterone, very low. So you have to put these charts with what's actually happening here. So now I'm at day 14. What happens at day 14? Ovulation. Y'all with me on that? Are we preparing the uterus for implantation? Sure. We're going into secretory phase. What hormones are high? Boom. Progesterone starts to rise. Estrogens are high. LH caused ovulation, so all of these are peaking except for progesterone. But now we start seeing progesterone come up, because what makes progesterone again? Corpus luteum. And you'll notice that the corpus luteum is not formed until day 16, and that's why progesterone starts to go up. You guys got that? Should you draw this out? Absolutely. Draw this out. This is a major chart. You see them in doctors' offices all the time, especially if, fertility clinics, because they want you to understand the importance of all of this and the days of the cycle. So you want to put all that down. I've got it in your notes, but I don't find the text notes to be incredibly helpful to you. You have to understand this as a 28-day cycle. So hormonal control. Small amounts of estrogen inhibit GnRH release through childhood. Then the hypothalamus is less sensitive to estrogen and begins releasing GnRH, gonadotropin releasing hormone. It's in your endocrine worksheet. This happens at about puberty. The process. GnRH release and stimulates the release of FSH and LH. FSH is in bold because in the first release during the beginning of the cycle, FSH, follicle stimulating hormone, is the important one, and it causes the follicle cells to begin to grow. FSH and LH both cause the release of estrogens. I've just laid it out for you numerically in 1, 2, 3, 4. We get negative inhibition with low estrogen. Low estrogen causes an inhibition. FSH is still present, causes more estrogen. Now we've got high estrogen. You might put in parentheses down here, high estrogen. High estrogen stimulates the release of LH. Now this is in bold. Circle this. LH causes ovulation. Cycle that off to the side. LH here in bold. Big jump causes ovulation. Causes meiosis to resume. Converts to the secondary oocyte. Ovulation occurs. LH causes the corpus luteum to release progesterone, double underline progesterone here, that's huge. It does inhibit GnRH. If fertilization and implantation occurs, HCG can be detected in the female bloodstream within 2 days after implantation, which is about 7 days after ovulation. That's how quick you can detect low levels of HCG. You go get a blood test for pregnancy at Walgreens, it's an HCG test, and it's a small chemical released from a fertilized embryo that's implanted in the uterus. And We can detect level changes within 7 days of ovulation, pretty quick. If you don't get HCG levels on that pregnancy test after 7 days, does that mean you're not pregnant? No, I mean, it's not, it's a false negative because it's too early. They usually tell you to wait until you miss a cycle or a menstrual cycle, and then you can test it and will be much more accurate.
But you said blood test, but you need urine test, right?
That HCG is in the blood, but it goes through our kidneys, which we now know about, and it is a urine test, thank you. It is found in the blood, will be transported, and that HCG test is a urine test for HCG in the blood. Yeah, thank you, Lindsay, good point. Uterine cycle then, can you follow this? What's going on in the ovary? What's going on in the uterus? There is correlation. Menstrual, proliferative phase. Menstrual, somebody give me some days of this cycle. If we're talking about a 28-day cycle, 0 to 5, menstrual, all right? Sloughing off, 0 to 5. Then 5 to 14, proliferative. 5 to 14. Secretory, 14 to 28. Caused by rising progesterone, arteries spiral in the endometrium, excess glycogen release. Why would we have glycogen in the uterus?
Energy?
Energy. These are developing embryos, implanting in the uterine lining. Need buffet. Ready for implantation. Degenerating corpus luteum causes sloughing of the cell. When progesterone levels drop, menstrual phase will begin and sloughing of the endometrium occurs. Questions on that? Here's what we're looking at for the final. I'll give you day 19. Day 19? You need to tell me about it. What hormones are elevating at day 19? Anybody know? These are the questions that come off the test, so I'm just giving them to you now. This is how you have to figure it out. Yeah, progesterone. Thank you, Liz. Progesterone's elevating right now. FSH, LH? No. They're going down. Estrogens? It's kind of about even up. Progesterone is elevating dramatically at day 19. Has ovulation occurred or is it going to occur? It's already occurred. So what is left in the ovary then? Corpus luteum. Corpus luteum, which is making progesterone. All right. What's the uterine wall look like? Day 19. Pretty thick. Secretory. It's ready for what? Implantation. Implantation. So you have to put it all together, see it all together. I'll give you days. You tell me what's going on in the ovary. Tell me what's going on with hormones. Tell me what's going on in the uterus. If you can nail those up just by putting a line here and seeing it all. Boom. Ovulation. What hormones are at their peak level right now?
That's all of them.
LH. LH. Because LH causes ovulation. That's what it does. How about progesterone?
No.
No, we don't even have any yet. A little bit. We don't have any yet because that comes from the corpus luteum, which hasn't even been formed yet at day 14. Can you guys see this? Not see it yet. At 2 weeks. 2 weeks. That is probably 40% of your final, is understanding how all these hormones interact with the ovary and the uterine lining. Complicated. Can you do it? Draw it out. Big drawing. I've already given you a drawing exercise on move. A little bit of work for yourself. Good. Questions on that? Questions on that? All right. Genetics, double line. Next topic. I gave you a terminology page on Moodle, you should print that out and you should be comfortable with it. If you had me for NV2, we already talked about phenotype, genotype, and we did dihybrid crosses and things like that, Punnett squares. So we're going to start kind of working toward our extra credit assignment. There's a couple tricky ones on there. Remember I said you gotta get them all right, so don't just rush through them. Check them with other people and make sure your answers are correct, and I'll be able to look at them in here on Wednesday and at least point out the ones that I see are incorrect, and you can work on them over the weekend. Many traits in our bodies are affected more than one pair of alleles. Alleles are pairs of chromosomes, one passed from the mom, one passed from the dad. When you say height, for instance, it's not just one set of chromosomes that determines height. Usually it's 3 or more. They call those allele pairs. Good example is blood typing. A and B are codominant. Height's another example I was just talking about. Talk about sex chromosomes, we talk about autosomal inheritance. Humans have 2 types of chromosomes, sex chromosomes and autosomes. So sex chromosomes, the X and the Y, and then everything else are called autosomes. So we talk about the chromosomes. Sex chromosomes, if you're XX, you're female. If you're XY, you're male. There are some conditions where we had non-disjunctions problems during fertilization. An XO would be Turner syndrome, that's 2n equals 45. Klinefelter syndrome is XXY, that's a 2n equals 47. Those are abnormal types of genetic situations. If they're not sex chromosomes, X and Y, the other 22 pairs are called autosomes. If it's dominant, autosomal dominant, it will be shown in the phenotype. It does not, you'll notice this does not mean that's good. Dominant doesn't always mean good or bad, it just means that if it's present, it will be expressed. Is that fair? Anybody know what the dominant number of fingers is? 6. 6. Most of us have how many in here? 5. That means that we carry the recessive gene. Is that bad? No, it just means that's how it's expressed. So you have to know these different situations. Couple disorders that you should be familiar with are Huntington's chorea, which is Woody Guthrie's disease, or achondroplastic dwarfism. These are dominant situations. So if we'll do an example of a Punnett square, if I told you that the mom was free from the disorder, but the dad had it, what's the probability their offspring have Huntington's chorea?
2%.
Can you see this in a Punnett square? Mom has it in a past one of her mom does not have it. She's autosomal recessive. If you figure out the Punnett square, you'll see that 1, 2 of the kids carry the dominant gene for Huntington's. So 50% of this couple's offspring will be having Huntington's chorea. What's that called? Homozygous recessive. Terminology. Print out your term sheet. That's homozygous recessive. What's this? Homozygous dominant. The only other one? That's heterozygous. Alright, so when I give you examples for the exam, if I say mom's heterozygous for Huntington's, dad is homozygous recessive, can you do a Punnett square and give me the probabilities? Can we do that? You guys remember this from blood typing? Alright, sort of. Dwarfism's the same way. So you're gonna have to think out dwarfism. That's a dominant allele situation. So can 2 parents who exhibit dwarfism conditions have children that are non-dwarfism?
Yes.
They can. How is that? Can you show me an example of how that's possible? Both parents express dwarfism. that means they have a big D. Is that correct? Because it's an autosomal dominant disorder. But they have a child who does not exhibit dwarfism symptoms. So therefore the child was this. Are y'all with me on that? So the genotypes of the parents, they had to be heterozygous. Of their offspring, what is the probability that they have children with dwarfism? 75%. 3 out of 4.
Is it true though with that condition, if they're homozygous dominant, it's being
Not anymore with healthcare anymore. It used to be a condition that was, the mortality was high, like Tay-Sachs or other conditions. But now with healthcare, even if you're homozygous dominant, you can survive. So 1, 2, 3, 75% of their offspring are going to exhibit dwarfism. But they can have a normal, non-dwarfism, normal. What would that be? Society calling it normal? Society normal, non-dwarfism child. All right. You gonna have to do these on the final? Sure. Autosomal recessive. Must be homozygous recessive. Examples of albinism, cystic fibrosis, Tay-Sachs, PKU. You guys ever heard of these disorders? This is what genetic counseling is all about. P-South, at least in the past, P-South over here at Riverbend has had 3 genetic counselors on staff because parents come in and they have a child who has cystic fibrosis. As a parent, would you like to know if you're going to have another child with cystic fibrosis? Sure. And so they'll go through and do these genetic tests, and you have to know that cystic fibrosis, for example, is an autosomal recessive disorder. So for exam, you're going to have to figure out which ones are the autosomal dominant or recessive, but cystic fibrosis. So 2 parents who were non-cystic fibrosis had a child with cystic fibrosis. What's their genotype? They're hetero. Mom's this, dad's this. They do not have cystic fibrosis, but they have a child with cystic fibrosis. And they come in and go, holy cow, I didn't even know that was in our genotype. Didn't know that was in our family history. What's the probability that we have another child with cystic fibrosis? And the genetic counselor would tell them what? 25%. Probability, single event. What's the chance that they have 2 cystic fibrosis children in a row?
20.25 to the
There you go. 2 events, they already had 1. Chance that they have 2, 0.25 times 0.25. So about 5%. Yeah.
Stan, is that supposed to be a little f on the top, on the right? Oh, I see, thanks.
About that. There we go. Probability, single event. Chance, multiple events. That's how we do genetics problems. Can you do this one? Do you know what heterozygous means? Is albinism a recessive or a dominant condition? Recessive. What's the probability that 2 heterozygous parents have an albino child? Can you figure that out? What do you think? Can this albino child have children with normal skin and hair color? Sure. Let's do it. Albinism. There's albinism. This means lack of pigmentation. all it is. Two heterozygous parents. Go with me on that? There's the child with albinism, 25% probability they have a child with albinism, 75% probability they have a child without exhibiting albinism symptoms. My next question is, can this child have children with normal skin and hair color? Sure. All they have to do is mate with someone who has the dominant gene. And the dominant gene will be expressed as long as they mate with someone who does not have albinism. They have a probability to have normal pigmentation. Children with normal pigmentation. Incomplete dominance. Heterozygous has intermediate phenotype, example of sickle cell. And then we get into one that you haven't talked about before, sex-linked inheritance. You have to set these up a little bit differently. And this is where we're getting toward your extra credit problem set. With sex-linked, you have to set them up with the X and the Y. If recessive, they usually show up on the male system. The two ones that I want you to cue into, you're gonna have to remember, when I say albinism, you have to remember that's a homozygous recessive condition. When I say dwarfism, you're gonna have to remember that's a dominant condition. When I say hemophilia, you have to remember that this is sex-linked. And this is where you use X and Y's. You don't use the F's and the H's anymore. You have to use X's and Y's. Hemophilia and color blindness. You guys are gonna figure out why I'm colorblind here in a minute. Y-linked inheritance, passed from the father to the son, and they're very rare. The only one they can actually figure out is hair on the ears is a Y-linked inheritance situation. So let's do a sex-linked hemophilia. Mom is a carrier for hemophilia. Here's how you set that up. She carries the hemophilia gene. You notice that's a mom, so it's XX, all right? Dad does not have hemophilia. That means he looks like this. When you figure out X chromosomes are big, Y's are short. Therefore, just because you have something on the H over here on the X, you don't even have it on the Y. So whatever mom passes you is what you're going to get. So what's the probability that these parents have a child with hemophilia? Can you draw it out? Uh-oh. Small h is hemophilia. What's the probability they had a child with hemophilia? 25%. What's the probability, trick question, so why it's on the extra credit, what's the probability they have a son with hemophilia? 50%. 1 out of 2. There's the sons. You guys see this? This is on your extra credit problem set. I'm doing it now to give you a point. There's the sons. These two are the boys because they're XY. You got that? How many of these sons have hemophilia? 1 of 2 of the sons will have hemophilia. So you see how things can be passed from the mothers then to the sons pretty easily? Because there's nothing on the Y chromosome to mask it. Draw this one out. My mom is not colorblind. My dad is not colorblind, but I am. X-linked, colorblind. We're right there, we're doing it right now. Mom's not colorblind. My dad's not colorblind. What are their genotypes? Mom's a carrier. What's that mean? X, big C, X, little c. Can you guys see this? Or draw out the square. It's on the final. What's my dad's genotype? X, big C, Y. You put that Punnett square together, you'll see in the bottom right corner, there's an X little c y, and that is me. Colorblind. Even though my parents were not. You guys see that? I'm seeing it. You'll need to for the final minute.
What does the big C and little c mean again?
Colorblind.
So big C means,
Colorblind, that means that you have normal genes, because this is a dominant. Colorblindness is only expressed in the small c. Good question.
But can you say that again?
So this is big C's. Big C's. I could usually make the little c like that. Color blindness has to be. Can women be colorblind? Yeah, but only in the XX little c genotype. It's rare, but it can happen.
So colorblindness is recessive.
Color blindness is recessive. Hemophilia is recessive. Let's do one. Let's do a problem. Dad's colorblind. Mom is a carrier. What's the probability that they have a colorblind son? Dad's colorblind. Mom is a carrier. What is the probability that they have a colorblind son? What's the chance they have 3 in a row? Doesn't get any harder on fun. That's a hard question. Probability they have a colorblind son. Chance that they have 3 straight. If you haven't had Punnett squares before, I don't know. Did you guys do this in AMP2, Jeff?
You didn't,
By one, okay. First thing you have to do is figure out genotype. So I said dad's colorblind. What's his genotype?
X,
X, little c, Y. Mom is a what?
carrier.
You have to figure out genotypes. Once you get the genotypes, it's easy. Said mom's a carrier. Dad's colorblind. That's a little c, big, this is a big C. So you do the square. Probability they have a colorblind son. There's the 2 sons, because these are the XYs. That's a big one, that's a small one. That is 1 of 2. Probability is 0.5 or 50%. Chance that they have 3 in a row. 0.5 to the third is chance. Take the probability, throw it into that equation. 25 times 25. About 5%. You guys got a calculator? It's Monday. 0.5. times 0.5 times 0.5, which I figure out to be about 5%-ish, 6%. Chance that they have 3 colorblind boys around. Well, I'm colorblind. That's me. Is my boy colorblind? What's the probability my boy is colorblind? 50%. He's not. Well, it's a 50-50. He's not colorblind. We good? You guys got that? I ask you to, first thing you do is you figure out whether it's a dominant or a recessive or a sex-linked disorder. Then you'll know how to set it up. Then you figure out the genotype, then you do your Punnett squares. We good? Take a break, 5 minutes, come back.